Здравствуйте. Здравствуйте. Меня зовут Нима Bakhtiari. Я являюсь PhD по фармакологии и преподавателем медицинского университета Jundishapur в Ahvaz. Прежде всего, хотел бы извиниться за то, что вы нужно отправить вам записанное видео и не могу присутствовать лично. I'm going to continue in English. Please accept my apology. I try, but I have some stress, maybe happen some misunderstanding. And that is my first time to want to speak in Russian and in a public place. I apologize about that. Okay. First of all, I would like to apologize for having to send you a recorded video instead of the attending in person. Circumstances have arisen in my country, of which you are likely aware. And that is we have found ourselves in a situation of the war and military tension. Nevertheless, our friends and neighbors in Russia have once again shown themselves to be a free and respected nation. Therefore, I'm very happy to present my lecture. Okay, the topic of my presentation related to the extremely important area of the research and development, involving in both academically and specialists from the industrial sector. It concerns the role of in silico study. When I selected my topic and my title of my lecture, I want to speak about in area of the neuroscience, but I'm trying to speak in wide. But my interest in area of neuroscience, and I'm experted in silico in area of neuroscience. Okay. Traditionally, scientific research is divided into two types of study: in vivo and in vitro. The development of the in vitro method emerged in a response to the several factors: ethical constraints when we working with the living organism, the high cost and duration of the experiments, and the impossibility of fully controlling experiment condition. In vitro experiment allow for much better control over research parameter, are faster to conduct and require fewer resources. However, the main drawback of the in vitro approach is that it does not reflect the complete function of the organism. Numerous cases have been described in modern scientific literature where the therapeutic agents show promising results under controlled laboratory condition, but failed completely during the testing in animal or human. After the pandemic, COVID-19, it became clear that the industry must rely on the new type of the research, computer modeling and analysis, we call it in silico. The emergence of the microprocessor and the subsequent development of the personal computer lead to the tremendous process in science. One of the most promising yet still understanding area is in silico research. This approach combines to strengthen both in vivo and in vitro method, enabling the creation of the mathematical model of the physiological and pharmacological systems and testing them using computer. They began it is essential to provide a clear definition of in silico. Okay, what is in silico? In silico refer to the conducting experiment and the modeling using computers and the computational models. This approach include the databases, data analysis tools, data mining, homology-based method, machine learning, pharmacophore modeling, QSAR, in abbreviation QSAR, quantitative structure activity relationship, and molecular dynamics. For explanation, I use a classification system based on the application area of this type of research. Other systems exist, but I prefer not to overwhelm the audience with the unnecessarily complex terminology. The application area of the in silico research including three main directions: drug discovery and drug design. That's the major type of this kind of a study. In silico clinical trial, and type C and C area, systems biology and disease modeling. First, I'm going to use in silico modeling. That is a very complex part of this area of the research. I'm going to speak first about the in silico modeling. What's the in silico modeling? I would like to introduce the general concept of in silico modeling. When we say in silico, we usually mean the use of the computer model to simulate a pharmacologic and physiologic process. In other words, instead of doing an experiment directly on a cell, tissue, or animal, we try to represent the system mathematically and then run the experiment inside the computer. Historically, our control experiment were mainly in vitro, in test tube or cells called cell culture, where we change the parameter at the time we observe the response. With the rise of the modern computing power, especially over the last 2 or 3 decades, we are now able to extend thousands of the control experiment into the digital space. We take what we want to know from the biology and pharmacology and translate it into equation and algorithms. And then use the computer as a very flexible experimental platform. The key advantages is that the in silico method can combine some of the strengths of the both of in vitro and in vivo. And in approaches like in vivo study, they can represent complex whole system behavior at the same time, and like in an in vitro study, they allow us to control parameter very precisely and repeat experiment quickly and safely. The combination help us to significantly reduce ethical issue, cost, and the time. Today, in silico modeling is widely used in pharmacokinetics. For example, predict the absorption, distribution, metabolism, excretion of the drug before the clinical trials. It's also very important in the study of the complex of the cardiorespiratory problem, where directed experimentation in the human can dangerous or simplify impossible. Simply impossible. So to summarize this slide, in silico modeling is not just drawing a graph on the computer. It's a systematic use of the mathematical and computational model to simulate drug action and physiological response in a way that complement and sometimes partially replaces traditional laboratory and animal experiment. What is the role among the classical research type? In this study, I would like to position in silico research among the classical type of the biomedical research. Traditionally, we distinguish between two main categories, in vivo and in vitro study. In vivo experiments are performed in a whole living organism, in animal or in human, and therefore they capture the complexity of the physiology, including system regulation, reflexes, and organ-to-organ interaction. However, they are expensive, time-consuming, associated with significant ethical consideration. On the other hand, in the in vitro experiments are performed in a controlled laboratory setting, for example, in the isolated cell, tissue, or the biochemical system. These experiments allow us to change condition very precisely and to study a specific mechanism in isolation. But there is an important limitation: in vitro studies often do not represent the behavior of the entire of the organism. The biological environment is simplified and many systems factors are missing. Okay, in silico research come in between these two classical approach and offer a bridge. In silico study, we construct a mathematical model of the physiologic and the pharmacologic system. This model can represent, for example, of the cardiovascular system, the respiratory system, or drug transport and metabolism in a quantitative way. Very important, in quantitative way. Because of all parameters and condition are explicitly defined by the researcher, we can simulate both local mechanism and whole body behavior. We can mimic an in vitro situation by isolating part of the model, or an in vivo situation by connecting multiple systems together. In this sense, in silico modeling help us to translate detail of the mechanistic knowledge from the lab to bench prediction at the level of the whole organism. So role of in silico research is to complement classical in vivo and in vitro experiment to integrate their finding in consistent model and to generate new hypotheses that can then to be tested experimentally. It does not replace traditional research, but it makes the entire process of the drug development and physiological investigation more efficient and more rational. In this, I'm going to say some example of the in silico modeling. There are many models, and I cannot cover all of them, and all of them are in formula, and I'm avoid to explain formula, and maybe some example to say important of the role of in silico in R&D. Okay. Ventricular interdependence. That is one of the model in silico modeling. And we see an example of how in silico modeling can help us understanding ventricular interdependence. Here the cardiovascular system represented by the electrical analog model in this framework. Maybe next, okay, that is a better presentation. In this framework, pressure and flow in the heart and vessel are mimicked using a component like a resistor, capacitor, and inducators allow to us to study the interaction between the right and left ventricle in very controlled way. The in silico findings are quite interesting. The model show that increasing the inner pressure in one ventricle can actually enhance the contractile capacity of the others. In other words, the two ventricles do not work in isolation. They mechanically support and influence each other. Moreover, simulations suggest that the right ventricular output can rise far beyond its own intrinsic capacity because of support from the left ventricle. This is a subtle phenomenon that is very difficult to isolate and quantify in vivo due to the complexity and the variability of the real patient. So, the example nicely illustrates how an in silico approach can reveal underlying mechanism of the ventricular interaction that would be hard or even impossible to study directly in animal model or in human subject. Another example of the in silico modeling, acute left ventricular failure. I will show how in silico modeling can be applied to very challenging clinical problem, acute left ventricular failure. A study of the severe acute left ventricular or LV failure in vivo is both ethically and technically difficult. In real patients, we cannot simply induce acute LV failure just to study the hemodynamic. Even in animal model, such experiments are highly invasive, risky, and often not fully representative of the human pathophysiology. This is make it hard to explore the full range of the condition and treatment option in the controlled way. This is this is where in silico model can be extremely valuable. In the computational model of the cardiovascular system, we can represent the left ventricle, the systemic circulation and the pulmonary circulation using a mathematical equation, and we have such a model. We can independently vary key parameters such as myocardial contractility, heart rate, systemic vascular resistance, or vascular compliance. For example, we can simulate what happens if contractility suddenly drop, or if heart rate compensate by increasing, or if systemic resistance change due to the vasodilation or vasoconstriction. We can systemically test many different what if scenario that would be impossible or unsafe to reproduce in a real patient. This allows us to explore the hemodynamic consequence of the acute LV failure in a very controlled and reproducible way. However, an important key message from this type of the modeling is that the results are highly sensitive to the choice of the parameter. Small changes in assumed contractility, resistance, or compliance can lead to the quite different prediction for pressure, follow, and volume. Sorry. This sensitivity is not a weakness of the approach. Rather, it highlights how crucial it is to carefully validate to the model and calibrate it using experimental or clinical data. So, the main point of this slide that in silico model provide a powerful, ethical, and flexible platform to study the acute LV failure, but their usefulness depend crucially on accurate parameter selection and rigorous validation against a real-world measurement. Okay, next slide. In next slide, in the that's the example. I will focus on the vascular system and relevance to the cardiac surgery. Large arterial tree models are a mathematical representation of the major artery in the body, including the geometry, elasticity, and branching structure. This model can use to simulate the different surgical intervention, perfusion, strategy before they apply in the real patient. One important application is the simulation of the aortic graft replacement in many surgery in a disease segment of the aorta is replaced with an artificial graft. These grafts are often much stiffer than the native aorta using an in silico arterial tree model. We can predict how these changes in the stiffness will affect the system and pulse, pressure, and all of them. Simulation of consistently show that stiff aortic graft tend to increase both mean systemic and pulse pressure. This has implication for the long-term vascular health for the risk of the complication, and another key application involve cardiopulmonary bypass or CPB perfusion strategy during cardiac surgery. Different perfusion strategy, such as varying follow rates, pressure, cannulation site, can lead to different distribution of the blood flow across the organ. With an artificial tree model, we can test this strategy virtually and observe how they change organ perfusion, for example, to the brain, kidney, or myocardium. The main benefit of the such in silico simulation is that they allow us to predict hemodynamic effect before the actual surgery. This can support the surgical planning, help to individualize perfusion strategy for a specific patient, and potentially reduce the risk of the adverse event. In summary, large scale of the vascular model serve as a kind of the virtual laboratory for a cardiac surgery where the impact of the graft properties and CPB strategy on the hemodynamic and organ blood flow can be explored in the safe and cost-effective way. Another example, mitral valve modeling. Here we move to a more detailed example of the modeling of the mitral valve. The mitral valve is the structurally complex with the leaflet, chorda, tendina, and dynamic annulus. Despite its local anatomical complexity, its function is the crucial for the global hemodynamic dysfunction of the mitral valve and can significantly alter the pressure and follow work in left atrium, left ventricle, and pulmonary circulation. In silico model of the mitral valve aim to capture both structure and its interaction with the rest of the cardiovascular system. Typically, this model combine in several component valve-specific equation that describe the leaflet motion and orifice aorta, time-varying elastance model that represent the pumping action of the cardiac chamber, and vascular model that can account for the systemic and the pulmonary circulation. When these elements are integrated, the model can reproduce the transmitral flow pattern, how blood flow from the left atrium to the left ventricle, and pulmonary venous flow pattern. Importantly, many these models show a good agreement with the Doppler echocardiography and invasive hemodynamic measure. This agreement provides confidence that a model can be used to study disease mechanism and therapeutic intervention. One key application is in understanding valve disease and expected outcome of the valve repair or replacement. For example, we can simulate how different type of the mitral regurgitation affect pulmonary pressure, or how particular surgical repair technique might change transmittal gradient and ventricular filling. Thus, mitral valve modeling is illustrated how in silico approach can bridge a detail of the structure, mechanism, chamber, function, and systemic hemodynamic. It offers clinician and researcher a powerful tool to analyze complex of the valve pathology and predict the hemodynamic impact of the different treatment strategy before they are applied in real patient. Okay, I'm going to illustrate docking and molecular docking and dynamic molecule, the most important role in research in the area of the drug design and drug development. That is an article and example that is I refer and suggest if you have a time, please study. They study in silico, and after that one or two in vitro study patch clamp together their work, and after that they're working on the patent. They catch a patent because of role of the molecular docking and small molecular docking and molecular dynamic. I'm going to say a very concise about molecular docking. Now I will switch from the organ level modeling to the molecular scale example of the molecular docking of the small molecule. Molecular docking is the computational method used to predict how a small molecule such as a potential drug bind to the target of the protein. The goal of it is to estimate the most likely binding pose, meaning the position and orientation of the molecule, as well as the pattern of the interaction, such as the hydrogen bond, hydrophobic contact, and electrostatic interaction within the protein binding site work. In practice, we start from the three-dimensional structure of the target protein, often obtained from the X-ray crystallography or cryo-electronic microscope, and a library of the candidate of the small molecule. Docking algorithm, they place each molecule into the binding pocket in many different orientation and conformation, and score each pose according to the chosen energy or affinity function. This approach is particularly powerful in early stage of drug discovery. Instead of the synthesizing and testing thousands of or million of components in the wet lab, and we can screen large virtual laboratory in silico and select a much smaller subset of the promising candidate. In this way, docking serves as a fast and cost-effective alternative to first round of the experiment assay. Of course, docking prediction are approximate and need to be validated experimentally. But even with this limitation, docking can greatly narrow down the search space and generate hypotheses about which chemical feature are important for a binding. Also help us to rationalize structure activity relation and design improve analogous. So the introduction of the molecular docking showed that how in silico method do not operate at the organ or system level, but also play a central role at the molecular level, supporting more efficient and targeted drug discovery workflow. Okay. A key concept of docking. On this slide, I want to introduce the essential concept of that from the function and molecular docking. These are terms that idea that we rely on whenever we use a docking as a tool in drug discovery, virtual screening, or structure-based design. First, let's talk about the ligand. In docking, ligand is the small molecule that we are testing in them. It might be potential drug candidate, a fragment, or compound from the virtual library. The goal it is to understand how this molecule might be bind to the particular protein target. Next, we have two receptor, which is usually a protein which a well-defined binding pocket. The receptor provide the 3D environment, the shape, the charge, and charge distribution, the hydrophobic and the hydrophilic region, and determine how we ligand can fit on that. In docking study, the structure accurately of the receptor is crucial because even a small error in the protein model can affect the predict binding pose. The third key of concept is binding pose. This is referred to the predict three-dimensional orientation of the ligand inside the binding pocket. Essentially, docking attempting to answer how does the ligand side inside the protein. The pose of including detail like the rotation, translation, and torsion angle of the ligand. To evaluate these poses, we use something called a scoring function. A scoring function is a mathematical model that provides an estimated binding affinity between the ligand and the receptor. It looks at the factors such as hydrogen binding, hydrophobic contacts, and electrostatic interaction, and something to fit into that. Okay, while the exact value of the are approximate, the score help us to rank the possible poses and describe which one might be represent a realistic interaction. Finally, a crucial binding entire the process is search algorithm. Docking is not simply placing the ligand once in the pocket and instead the search algorithm explore many possible conformation and position. Some of the algorithms systematically sample the torsion and orientation, and while the other use of the stochastic approach like a generic algorithm or Monte Carlo method to efficiently explore the space of the possibility of that. Without the good search algorithm, even the best scoring function cannot finding the correct pose. So overall, this slide summarizes the core of the element of the docking, the ligand, receptor, binding pose, scoring function, and search algorithm to all process of the docking. Together, they allow us to simulate and analyze a protein ligand interaction and support information decision through the drug discovery pipeline. Okay, in this slide, I'm going to speak about the docking workflow. This slide outlines the typical workflow used in a standard molecular docking study. Even through the different software package implement this step in a slightly different way, the general sequence remain very consistent across the tool. The first step is the protein preparation. Before the docking, we need to clean and usable structure of the receptor. This usually involves removing the crystallographic water molecules that are not functionally important, adding missing hydrogen, assigning a proteination state for the key residue, and fixing a structural issue. A well prepared protein is essential because the quality of the input directly affects the quality of the docking result. The second step is ligand preparation. Just like a protein, the ligand need to be processed. We need to assign the correct protonation and tautomeric state. Because of this strongly influence hydrogen bonding pattern, and we also perform energy minimizing to ensure the ligand start from the responsible low energy conformation, and many docking error actually targeted to the poor ligand preparation. The next step is grid generation. Here the software define that the region around the active site binding pocket where the ligand will be tested, and the grid essentially encode the receptors shaped and the energy land space, making the docking process fast per computing interaction potential. Then we reach the central step, the docking ground. This involves the simplifying many process binding process and scoring them. The search algorithm explore the different orientation and conformation and the position of the ligand. Each sample pose is the evaluated using the scoring function. The output usually a list of the rank pose from the most of the favorable to the list. Finally, we have a post processing. This is where we interpret the result. We examined the candidate process, visualize how the ligand fit. I think that is enough. On this slide, I would like to focus on the central component of the any docking protocol, the scoring function, and the main limitation that come with that. In a molecular docking, a scoring function is essentially is mathematically model that tries to estimate how strongly a ligand will be bind to its protein target. In practice, we use a different type of the scoring function, some of the empirical, where the parameters are fit against the experimental binding data. On other are knowledge base built from the statistical analysis and many non-protein ligand complex. These are also force field based scoring function that rely on the classical physics summing up van der Waals and electrostatic terms. More recently, machine learning enhanced scoring function have been popular, using data-driven model to improve the predictability of the binding affinity. I'm going to speak about the molecular dynamics. The molecular dynamics is very important. I'm pass away some of the part of my slide. As a result of the docking, scoring always comes with the degree of the uncertainty. This is why any serious docking study must involve validation. We typically compare docking result against the known experimental structure or binding data, or we often complete that with the more detail of the method, such as the molecular dynamics simulation. Molecular dynamics can reflect the system's incorporate protein flexibility and provide a more than realistic view of this complex over the time. So the main message of the this slide is a scoring function is very useful for fast screening and sometimes remain approximation of that in all of that. Okay, We should always be aware of their assumption and limitation of we should use experimental data or more advanced simulation to validate or refine our docking. This slide is about the application of the docking. Maybe application of the docking is very important, but I will highlight the main application of the molecular docking is modern drug discovery and the chemical biology. And one of the most common use of the docking in the virtual screening and large component of the library, instead of the physically testing the thousand or million of the molecule in the laboratory, we can computationally dock them into the binding site of the target of the protein. The scoring function then allow us to rank the compound and select the relatively small and subset with the highest predict affinity for the experimental testing. Because of I'm going to, I did not want to miss any part of my speak. I speak completely, but I can pass away this to a slide. That is, oh, that is my important note. The every day technology work, even we don't not fully understand. Like a microwave, we push a button and warm food, but we don't know how. We have a new player role in modern area in recent area of the research, and that is the AI. AI help us develop our R&D and use this mathematical and very complex formula simply. The goal of our presentation, develop the Russia and Iran framework for the industrial and scientific AI research, and I wish we can develop that. That is my phone number. Have a WhatsApp and Telegram, but by the way, in area in the time of the war, the Telegram and WhatsApp are filtered, but my email is available, m_nima_b@yahoo. That is my first email that I check it every day, every 5 minutes. And if you send me an email, I will be able to I will connect you and be I'm very glad in Russian очень рад to познакомиться with вам. Okay, thank you. Please accept my apology. I'm going to speak more in detail, but they were very important. I wish it is a good presentation and I didn't miss anything. Thank you.